now coming to the concept of the microbiomes. So what is a microbiome? Microbiome, this was in fact actually coined only very recently in 2001. That means previously we understand in most of the cases, even now also the belief is the microbes are very bad. Microbes are very bad because we now understand, now the whole globe understands the ill effects, bad effects of COVID. So therefore most people talk about the microbes are very bad. But if the microbes are very bad, we can't survive. Very few microbes are bad. Very few microbes are bad, and most of the microbes are. So there's a good, bad, and neutral microbes exist everywhere. And microbes are ubiquitous in nature. We find in water, air, everywhere, food. But these microbes, when they are associated with a habitat or with an animal or a plant, collectively all of them called microbiomes. So this is a simple definition, the set of microorganisms of a particular habitat, or the set of genomes of the microorganisms in a particular habitat. So that means if you talk about easy definition, human being. So humans, people popularly now considered as 90% microbial, 10% only we are human in terms of the number of cells, not by weight. So we are more of microbial because we have the viruses, bacteria, fungi, and also microscopic fauna. That means we are more of microbial than the human. And all these microbes associated with the human, wherever they are, human microbiome. If the microbes of the skin microbiome, nose, nasal microbiome, or gut microbiome. So likewise, including vaginal microbiome. Now, this is growing and a frontier science across understanding the relationship between these microbes and the human behavior, human mood swings, and human requirement, every activity, human metabolism is under the influence of the various microbial communities. Likewise, even in the plants, we have these microbiome communities. They inhabit the leaf or a stem or even inside the vasculature or at the rhizosphere or maybe the surface of the rhizosphere, all collectively called as plant microbiome. And if the plant is a crop microbiome. So what is this microbiome? How many types of microbiomes are there? How this microbiome influence the plant? So therefore, we cannot actually survive without the association. That is what we understand over the few decades. So these microbes, culture dependent or culture independent. We can only culture 2 to 3% of the microbes. Rest of the microbes we can't culture as of today, but we can understand the microbial communities based on the metagenomic analysis. So therefore, the microbes are very important and prominent in the life cycle of every living creature. That's why this microbiome is called extended genome or second genome of the plants and animals. Hope you understand what is microbiome. So now, how to harvest or harness the potential of microbiomes? Are you able to see my slide? Hello?
Yes, sir.
Okay,
yes, sir, we can see.
Yeah. So the fundamentals of microbiomes. So they are complex and diverse communities of microorganisms, including viruses, bacteria, archaea, and fungi, etc., etc. Most of the microscopic life forms. The diversity, the complex interactions, the functional roles, associations with the host, the resilience, and they render ecosystem services. That is the very, very important component to understand. Sometimes we can't dictate these microbes and their functional abilities. And these microbial services even without asking they does. Likewise, there are ecosystem services rendered by several insects. The biggest, the ecosystem service rendered by the bees, nothing but the pollinations. Today, we are surviving because of the presence of the bees and other insects, which involves in population. That is, are you asking? They are doing it as a service. So these ecosystem services, every organism, every maybe an insect or animal that does ecosystem service, whether recognized or not. But now coming to the diversity, of course, there is a lot of information is there, but I think time is already 12:06. How best these fundamental systems we can utilize to improve the agricultural productivity in a sustainable manner. That is the goal. Let us move forward. So, one spoon of soil microbiome consists of 1 billion individual microbial cells, around 10,000 different species. You see the diversity. 10,000 species in a spoon of a soil which is having a 1 billion individual microbial cells. So this is the diversity. And do you think all these are pathogenic or maybe useful? Neutral, as I said, very few are pathogenic, and maybe considerably more are good, and 99% are neutral sometimes. But what we are now looking at, both those are pathogenic to the plants and also useful to the plants. We have to look at both the sides and how to bring balance between good and bad, and how to impart more good into our agricultural system, and what are those microbial technologies today available for us. The diversity we are now talking about, we'll go till deep into understanding the diversity, especially with regard to the plants. So I mentioned the soil microbiome. It may be the loose soil or maybe the soil around the root system. That's generally we call rhizosphere. And those microbes which are present inside the plant, that is a vasculature or the leaf or root or stem, we call them endosphere. And those microbes are present on the surface of the leaves and other parts of the plant. That means above ground, we call them as phyllosphere microbe. And also the airborne and waterborne, the microbes are there, maybe air microbiome or water microbiome. So as I mentioned, already defined the microbiome. So holistically, we can call it as a microbiome or crop microbiome or plant microbiome, but can be subdivided based on the presence. So if it is a rhizosphere or phyllosphere or maybe rhizoplane or maybe the what you call bulk soils. So there is a huge diversity of these microbes in not only the presence but also the different kinds of microbes. As I mentioned, there are plants which are diseased, which are under impact of pathogenicity. Those we call them plant pathogens like root rot, dieback, and damping off, wilt. So apart from that, we have the anthracnose, we have the viral diseases, fungal diseases, bacterial diseases, and at the same time, there are innumerable number of the various microbes which are very, very important that we do not know 15-20 years back. We know only very few microbes like rhizobium, nitrogen fixation, azoospermillum, azotobacter. So like that, very few microbes we know they are important. But today, thanks to omics, and we understand the each and the function, every function of each microbe, how these microbes are associated with the plant, at what level, physiological, biochemical, or genetic level, their associations, actions, interactions, now we understand thoroughly to the core. This is the plant microbial world. Now we are going more into the plant side. And there are several kinds of the microbes, right from the nematodes to algal to bacterium, all microscopic in nature. But today our focus is mostly on the bacteria and fungi. So this is where actually a plant showing both the effects of the harmful microbes or the beneficial microbes. But today we are talking about the microbial technologies and the microbial systems, the microbial communities help in agriculture productivity. There are symbiotic microbes, plant growth promoting rhizobacteria, the endophytic microbes, various biological control agents, and also the beneficial viruses. And there are so many microbes involved in nutrient use efficiency, nutrient recycling, and absorption, assimilation, fixation, mobilization. So these are the various functions rendered by the microbial communities, which are, of course, not visible, not known to us before, but today we understand each and every step involves in these nutrient recyclings to the induction of defense mechanisms in the plants. Now coming to the microbiomes associated with the plant, start from seed microbiome. So this is another wonderful subject. It's a lot of research is happening. We know all these years, the breeding, the seed production, F1 hybridizations, all these things at the breeding level. Today, the scientists understand, and they really now thoroughly convinced that the microbes associated with the seed also play a role in crop productivity. The microbes, that means the seeds, they pack their microbes. And these microbes, microbe in the sense is not just like a microbe, it has its own genes, the functional genes. That means here, the characters of the parent to offspring also through the microbial integrations of the microbial genes. That is, now we understand that's why the microbiome assisted plant breeding is also shaping now. So microbial seed endophytes have an advantage over soil bacteria during plant colonization. So that means we know the bacterialization of the seeds, we know the seed priming, seed coating, seed treatment, but at the same time, more than that, the seed endophytes play a major role in their survival and also absorption of the food materials. So therefore, there's a lot of research is happening to understand seed associated the microbial communities we popularly call seed endophytes and their role in metabolism. So during seed germination and increasing the seed vigor and seed growing seed germination or germinating seeds, they withstand the abiotic and biotic stress conditions. Abiotic, the drought and salinity conditions, and biotic, maybe the pathogen effect. So therefore, seed microbiome is the one. And are you able to see rhizosphere microbiome?
Yes, sir.
Rhizosphere microbiome, are you able to see the slide?
Sir, it's on seed microbiome.
Agreed. There is a problem. Now?
Yes, it's visible, sir.
Yeah. So rhizosphere microbiome, the rhizosphere is nothing but the active zone, very active zone of the rhizosphere, that means a root, and it is highly colonized by various kinds of microbial communities. See, it is unimaginable kind of a dynamism you see around the rhizosphere. It's a very narrow region soil that is directly influenced by the root secretions. These root secretions, we call them as root exudates. I have another 2-3 slides focuses on this because this is very important and vital. What do you call the functional phenomenon exhibited by various microbial communities and their interactions with the root systems. So therefore, the rhizosphere is very vital in terms of their innate immunity and their absorption of the nutrients, assimilation of the nutrients, and also recruitment of various kinds of microbes. I will show it in a future slides. So therefore, rhizosphere microbiome plays a really great role. So the optimization of the rhizosphere is now the future trend. It's already a trend, and the future we are going to have tailor-made formulations to optimize the rhizospheres of various crops. That means crop-specific rhizosphere optimization. It is really going to be a wonderful phenomenon, and also a lot of new delivery systems will be available for individual crops to help the plant to survive and abiotic stress conditions and biotic stress conditions, and also the nutrient utilization. So this is a philosphere, and that means the microbes inhabit the surface of the leaf. They play great role. It's highly dynamic in nature, exposed to the atmosphere, not like rhizosphere. But it is largest several kinds of the microorganisms, which interactions are very essential for the plant for absorption, and also involves in photosynthesis, and also the stomatal movements. There are many things. So likewise, so we have now the various microbiomes like endosphere, and we have insect microbiome also play major role. Then that means seed endophytes or seed microbiome, soil microbiome, endosphere, philosphere. So it's all depends upon the various kinds of the microbial communities which take part in association with the root system, or maybe the vasculature or surface of the leaf, or maybe you will find a different kind of communities in the bulk soils. So therefore, we totally understand today the intricacies and also interactions between these microbial communities and the plant at the molecular level. So now, having known the functions of these microbes and their role in plant life, and we have few sets of microbes identified, and we call them as agriculturally important microbes, AIM. So these microbes involves in right from carbon sequestration and also the insect pest control. As I mentioned about insect microbiomes, so likewise you have the entomopathogenic microbes like entomopathogenic virus, entomopathogenic bacteria, fungus, and nematodes. So they are not associated with the plant, but they are associated with the insect, and which will control naturally the insect populations. So they play a great role in crop protection. But nevertheless, today we have agriculturally important microbes. Is a huge list of microbes available now, and how these microbes involves in the various kinds of activities. These activities, we know the actions and interactions, or maybe the direct or indirect actions. So you can study all these things using, I was mentioning about the omics, we call them microbial toolbox. The microbial toolbox, right from starting with the culturomics to phenomics, genomics, genomics, transcriptomics, metabolomics, proteomics. These are the various platforms today readily available to understand plant microbial interactions. and microbe interactions. And precisely now you can have a microbe which has a single or multiple functions. And there are analytical methods. I mentioned about the several analytical methods, right from the physiological to the molecular things. And now these are the direct actions, indirect actions, and the interactions. So these are the microbiome actions and interactions. And each action is very important, and each action you can trap, you can find out or identify a consortium or a single microbe, you can convert into a delivery system. In the sense, you can develop into a product with precise actions. Of course, a lot of technology, infrastructure, knowledge, all ready, not are required to have to produce these target specific delivery systems. And these are the same thing which is depicted in the form of the picture here in the design, where you have a lot of mutualistic, the mutualism is there, symbiosis is there, antagonism is there. So these are the various the functional phenomenon exhibited by these microbial communities. So now let us start understanding the role of microbiomes at various levels. The first come first is the soil. Good soil means the 5 important components: soil organic matter, soil minerals, the water holding capacity, the aeration, and the microorganisms. Today we are now talking about the microbes, and these microbes are very, very essential to build organic matter in the soil. The microbes are very, very essential in soil organic matter recycling. So I have shown you already rhizosphere. So these microbes, they help especially to enhancing soil organic matter, even they are alive or dead. So that is the wonderful gift to the mankind, the wonderful gift of the nature, the wonderful microbes. They help us while living or even dead. So while living, they helps the plants in many ways, and of course, even human beings. So the rhizosphere is very active. I told you the very active zone, and the zone of soil under the direct influence of the plant, because the plant roots produces tremendous exudates that attracts the various microbial communities. There is exchange of information, exchange of nutrients, exchange of signals. And deuterosphere, that is another thing where the microbes, once they dead, they form dead decaying organic matter that is nothing but the humus that holds a lot of moisture, that holds organic carbon. So therefore, the microbes, dead or alive, they help us in many, many ways. And these are very, very important essential components in soil regeneration to capture carbon. So nowadays, the decarbonization, the carbon sequestration, especially soil organic carbon sequestration. So these, every company, everyone talks about how to remove the carbon from the atmosphere, send into the soil or into the oceans. Of course, oceans are not easy for us, but we can definitely, we can fix atmospheric carbon into the soil through the plant and microbial, what we call components. And carbon sequestration, yes, the plant, the microbes, both are very, very important components to sequester the carbon, and I have discussed that. And coming to the soil microbiome management, now the lot of work is happening across the globe, and especially working on soil microbiome management with respect to the individual crops, with respect to the different kinds of soils. And regenerative agriculture, especially what you call focuses on the soil microbiome or soil health or soil fertility managements, where it is very important, as I mentioned, about the crop rotations and also intercropping and less tillage. and also the keeping the biodiversity on. So if you can practice these things, definitely you can restore the soil fertility. And rhizosphere microbiome, I mentioned already all these things, right from the mycorrhizal fungi. Of course, now the farmers understand the importance of the mycorrhiza, not only the mycorrhiza, other plant promoting microorganisms or PGPs, so plant growth promoting microbes. It may be bacteria and fungi, and a lot of companies are now coming, and every international company, every multinational company today, they want to have biological products in their basket. So they all understand now, yes, the importance of biological agriculture, the importance of natural farming, the importance of organic farming, the importance of reducing the chemical load in the environment, and the importance of the immunity, the importance of the productivity, and importance of the soil fertility. Therefore, yes, how to bring balance between the chemicals and biologicals. So therefore, every multinational company or Indian big company, every company is now looking at the biological alternatives, especially science. So therefore, today you understand today now, I think these microbes play a very essential role, and lot of activities. We know very few activities before. Now we understand the each and every minute activity of the microbes, which helps in enhancing crop productivity, and that leads to the food security and sustainability in agriculture. And plant root exudates, I mentioned, this is very, very, very important. The root exudates, see, there's at the different stages of the same crop, the plant root produces different kinds of exudates. And it all depends upon the metabolism of the plant. So likewise, during seed germination, depends upon it is oil seeds, or maybe cereals, or maybe pulses, the exudates composition changes from plant to plant, dicot to monocot, or maybe the oil seeds to the pulses. So it all depends, once again, the exudates released by the root system. Those exudates has capacity to attract certain communities of the microbes. They may be beneficial or sometimes pathogens. So understanding the root exudates of individual crops is now becoming a very important factor to integrate this science in crop production. And as I mentioned, the rhizosphere is that recruits the essential communities. That means it's a recruitment means, so that means the set of microbes which are attracted towards the plants based on the exudate compositions. That is, now we need to understand thoroughly. So what exudates preferably attracts the good microbes, which their association is very important for survival, growth, and productivity of the plant. Now today, tomorrow we can have genetically modified a crop which produces a set of microbial exudates which has micromolecules which attracts very important microbes. The future is not that far. We are going to have this kind of a precision-based agricultural activities and research. So likewise, nutrient cycling. I mentioned the different types of plant communities produces different types of exudates. They attract different kinds of microbial communities. So likewise, these microbial communities play a great role in especially the plant health, soil fertility, and nutrient recycling and nutrient mobilizations. So therefore, these microbes, that means we are talking about the microbial communities which are essential for the plant survival, growth, development. That means in the yield. So there are several microbes, and their functions are nutrient exchange, phytohormone production, and induced systemic resistance. This comes through the mutualism. That means interdependence between these microbes. What are those microbes? Right from endophytes, rhizobium, and also the mycorrhizal fungi. And as I mentioned, every metabolic activity of the plant is sometimes governed or under the influence of microbes. It may be the microbial interactions or microbial exudates or the microbial signals or microbial metabolites. It can be hormones or gibberellins or many other growth development components. So here it is listed, but we do not have time to discuss another things. That means we are going deep in understanding the microbial interactions, what kind of their associations influence the plants. So today plant is not alone. It has an excellent associations with the microbes. We don't think of these good microbes. We always talk about the pathogens. No, the other side of the coin is very bright, that there are hundreds and thousands of the microbes which involves in day-to-day the functional activities of the plant. So it is noticeable to us, but now today, thanks to the technology, we understand how important these microbial communities. Now in future, now next few slides, what are those microbes, how to trap these microbes, how to understand these microbes, how to convert that into a product, and how to give this product as a delivery system to the farmer to manage his crop. That we are now going to talk. Yes. Now coming to the other aspect. So we call engineering of the microbiomes or optimization of the microbiomes. Here engineering, the term is used to say about assembling of newer communities, assembling of the essential microbes. So what are those essential microbes? Why they are essential for the plant? Now we are talking about the plant. We are talking about improving the crop productivity, and we are talking about the mitigation of abiotic and biotic conditions using the microbial delivery systems. So the manipulation of the rhizosphere or manipulation of the philosphere, or you can also manipulate the endosphere using synthetic communities or maybe the natural microbial communities. This we generally call as microbiome engineering. So this is just like a technology platform, a schematic representation, where you have to have your functional microbial bank. So this bank you develop using your omics platform or metagenomics platform, whatsoever, where you have a deposit of various kinds of microbes or microbial gene pool which are tested for specific functions. What are those functions? We have already discussed. It can be carbon sequestration, mobilizations, fixations of nutrients, or induces systemic resistance suppression, insect control, or immunity, and hormone production, whatnot. So every function you must understand thoroughly at the molecular level, and you can store those microbes under FMB, functional microbiome. And these microbes or microbiomes are from different ecological niches. I said it need not to be always from the cropland. It can be desert ecosystem, or maybe marine ecosystem, or a grassland ecosystem, or a forest ecosystem. But a diversity of microbes, bring it to your lab, understand and their functional capabilities, and study them at different levels. The primary screening, secondary, tertiary, greenhouse, at the field level. Then you can deposit that at the different levels. There is another science again, but you can have a bank of microbes, or a repository of the microbes, where you can convert these microbes into a delivery system. It can be a powder, liquid, granule, tablet, or injection, which can be administered. You can use for seed treatment, soil applications, foliar sprays, injections. So these are all the novel delivery systems available today. So that means now the circle is complete. So right from understanding, characterization, functional studies, and now the tools are available. And every activity, I was mentioning there are several activities, every activity can become a product. Every product can be a component of your business. If you are into research, yes, there is a research aspect, or if you are into business, it is going to be a business component. And this is what we have in our own laboratory, what we do, the microbiome approach for drought mitigation. So it's only just a simple example. So we have selected microbes, which involves in exopolysaccharide synthesis, IAA production, or maybe salicylic acid or jasminic acid production, ACC deaminase, or antioxidants or osmoprotectants. You know what is happening once the drought is onset. So there are several biological and physical changes happens, metabolic changes happen that should be intersected using these microbial communities. Each one has a different function, so I can't explain right now. But if you have a consortium of microbes which are tested to mitigate the drought, then you can integrate that through a seed treatment or a soil application, thereby to protect the root system of the crop, and it helps the plant to withstand the severe drought situation. This is only schematic representations. And these are the, how do you construct such kind of a formulations? Yeah, I mentioned here the selection, the compatibility, and assembly of the consortiums and inoculations. I mentioned about the 3 or 4 types of the ways of integrating through seed treatment or injections, foliar sprays, or rhizosphere optimizations, and then followed by the metagenomic analysis and the monitoring throughout the life cycle of the plant, and also regulatory compliance. So I will come to that later. Regulatory, what is regulatory compliance in using microbial technologies in agriculture? So this apart, today we talk about synthetic biology. So the microbiome engineering, sometimes it can be natural or maybe a synthetic in nature. That means the diversified communities you can bring, and you can have a synthetic microbial consortia. It need not to be always genetically engineered. That is a different story altogether. But today you have the tools. You have the tools to understand the individual and also the consortium and the functional abilities. So you can have today a synthetic microbial communities, but the only thing is this synthetic microbial community can adapt the local conditions, or maybe through seed bacterization or seed pelletization or rhizosphere optimization. But today, yes, you have the ways and means to understand the microbial communities. There are several technologies available today to make this microbiome formulations more functional with more shelf life and more vigor. So therefore, these tools like fermentation. So where you have the upstream and the downstream. The upstream mostly you can have a metabolite production, or you can produce the microbes, and downstream you can have the various kinds of methods like a freeze drying, spray drying, vacuum tray drying, centrifugation, separation, the volatilizations. There are many downstream processes are available, technologies are available, equipment are available. It is not a rocket science, but only thing is right from proof of concept. That means a discovery to delivery. Today, thorough protocols are available. Therefore, understanding the microbial abilities and understanding the plant requirements, how to match between these two, where we started talking about the dysbiosis. So we need to break that barrier and so make available these useful microbial consortia to the plants, and where they are really looking for their what do you call associations. And coming to the applications, yes, so we have a regulatory framework everywhere, even in India too. So microbial technologies, especially for agriculture, so falls generally into 2 categories. One is fertilizer category, another one is pesticide category. So that means those microbes used for fertigation of the plant, they are comes under fertilizers. It is governed under FCO, Fertilizer Control Order. And where those microbes used for crop protection, they are under Insecticide Act, IA 1968 and FCO 1985. So these are the 2 acts which governs all microbial products in India. So especially under FCO, Fertilizer Control Order, so we have biofertilizers, microbial biofertilizers, and microbial biostimulants. Whereas biopesticides and biocontrol agents, they are governed under Insecticide Act. These are the two acts. So biofertilizers, there is a clear difference between biofertilizers and biopesticides. And biofertilizers, usually they never carry any kind of nutrient, but they are the microorganisms. So they helps in fixation, mobilization, solubilization of various kinds of nutrients, and thereby the plant will be absorbed or supplied. And at the same time, we have another group of microorganisms now popular. They comes in biostimulants, because the biostimulants is the indirect action of the plant. Microbes. They are all called as PGPRs, plant growth promoting rhizobacteria. But these is a wonderful group of microbes. It may be bacteria, sometimes plant growth promoting fungus also, that involves in the various activities, especially the nutrient uptake and conferring the abiotic stress tolerance. That is very, very important in the current context. There are several kinds of species, like Bacillus is the most dominating one, followed by Pseudomonas, Azoospermillum, Azotobacter, Burkholderia. There are many, many kind of microbes which are now grouped under plant growth promoting rhizobacteria, plant growth promoting bacteria. It need not to be always rhizo. It can be endophytic or phyllosphere microbes. So there are big group of microbes are there. Biopesticide I already mentioned about. So right from entomopathogenic viruses, NPV viruses, Bacillus thuringiensis is a bacteria, and entomopathogenic fungi like you have Beauveria, Verticillium, Metarhizium, Phacellomyces. So likewise under biocontrol agents, especially for the biofungicides, also comes under biopesticides like trichodermas, very popular, and Pseudomonas are there, and Bacillus strains are there. They all comes under biopesticides. And the decomposing organisms, you know pretty well now, this is a very, very important initiative of ICR and IARI. They developed, and every agriculture university is coming up with their decomposing organisms. They to very properly decomposing solid organic waste. It can be a city waste, or it can be kitchen waste, or it can be a farm waste. So you know what is happening in North, especially Punjab, Haryana, where the stubble burning. Now, therefore, the government is coming up with biocomposting of the stubble, the material residues and other things that can be effectively converting into very important what we call compost that will help in nutrient cycling. And inoculants, yes, as I mentioned, they're all nothing but the biofertilizers again. So, and microbial biostimulants, I just mentioned the difference between biofertilizers and biostimulants. Biofertilizer, they fix a certain amount of nutrients. It may be N, P, K, zinc, or anything. But biostimulants, mostly their actions are indirect. They help and they trigger the metabolism of the plant by secretion of the various kinds of nutrients, or secretion of various kinds of hormones, or maybe stress mitigating hormones. Soil microbiome management, we have already discussed about it, and the precision agriculture, and the microbial biocontrol, that is nothing but we are talking about the biocontrol agents of Trichoderma, Pseudomonas, Beauveria, BT, Verticillium, Metarhizium. These are the only today available for us in India, unless until somebody comes up with genetically modified, that is agreed by. Unfortunately, not even microbial consortium is not introduced, but there are certain guidelines. But it's a fundamentally a business activity. One needs to understand the regulatory framework of this country, and we need to develop the products as per the guidelines of the government of India. So now coming to the very important activity is the soil microbiome analysis. Yes, Government of India initiated soil health card that only talks about the physics and chemistry of the soil health. But now across various countries, US and Europe, and even South America, the biological tools are available, especially the metagenomic tools are available to understand soil diversity, microbial diversity. That is very important. Based on that, so now the tools and diagnosis is available to understand and prescribe. Decision making can be done based on the microbial diversity. Whether your crop is going to get a disease, you can predict, and what is actually soil nutrient levels, what kind of microbial communities you require to introduce to properly manage the nutrient state of the soil. So likewise, the prediction models are coming up based on the soil metagenomics. And crop specific, I mentioned about the root exudates, understanding the crop specific microbiomes, understanding the seed microbiomes, understanding rhizosphere microbiomes. This science is growing, and maybe products will come into the market in next decade, and very precisely and going to form a part of precision agriculture. And seed treatment, yes, the delivery is always prophylactic in nature. The seed treatment gives more cost benefit ratio to anyone. We have proved that in several cases. So if you can start using the seed treatment, biological preparations as inoculants or maybe seed priming, so definitely helps in enhancing the vigor, the seedling germination, percentage of the what you call seed germination, and also they can withstand abiotic stress and biotic stress. And there are several microbiome approaches are there. So these are the biofertilizers, bioinoculants, and also the various kinds of preparations are available. I have mentioned about the various kinds of formulations. The formulations may be seed treatment formulations, soil application granular formulations, or foliar formulations. Even today, the tablets, capsules, and injections are also available. But there must be very important a caution for everyone. This is you are dealing with microbes, which are the living entity, living components. Therefore, we need to really respect so their shelf life, their mode of action, their food habits. We need to understand each microbe thoroughly in terms of their physiology and life cycle. And therefore, we need to give them very proper conditions for their preservation and also further growth and development. Therefore, the concepts of the prebiotics, probiotics, and postbiotics. Of course, this is another subject, but here in this case, what is most important is the discovery to delivery, where then only your real microbial technologies will see the light. So formulation is very critical, and when you are using a consortium of microbes, the compatibility is very important, whether they can compete with the local microbes. If you are introducing into the rhizosphere, introducing to the seed into the soil, already there are millions of microbes present already in the soil. Whether these microbes can compete with these populations or the survival of these edaphic conditions, that means soil conditions, soil temperature, soil moisture, soil organic carbon, soil nutrient status. So these are the various factors you need to consider. I mentioned here soil moisture and soil organic content, etc., and also the donors. I told already there are several formulations, several technologies available through downstream processing. So therefore, one need to understand the challenges in application of the microbial technologies is not just like a theoretical science, it's an application science where ultimately either you will be giving an advice to the farmer or a product to the farmer. These are the only two. Soil testing, seed testing, or plant testing, you can understand the microbial interference and microbial compositions, where based on that, you can give an advice to the farmer. And at the same time, where you can give a product to the farmer, either for the seed treatment, soil application, or foliar sprays. So I have already mentioned about these regulatory components. So we have biopesticides, biofertilizers, and the microbial biostimulants. So where today you can apply this microbiome concept or microbial technologies for seed health, plant health, crop yield enhancement, disease control, and all leads to the sustainable agriculture. Then coming to the business components, I have already mentioned about the seed priming, soil amendments, and foliar sprays. And finally, we'll be having like a seed vault. You know, the seed vault, the northern hemisphere, all the germ plasm which is available across the globe, all stored. If at all any catastrophe happens, there is a availability of the seeds for the humanity. Likewise, we can also build the microbiome vault, all the essential microbes where the microbes are very important. Those microbes can be stored in the microbiome wall as a functional microbial bank. So this is what we have created a concept. It is called MAA, Microbiome Assisted Agriculture, where our focus is seed, soil, and plant health, especially helping them from nutritional, abiotic, and biotic stress, especially taking the microbes from rhizosphere, phyllosphere, endosphere of the plant. So now everyone talks about One Health, but we propose one soil, one health, because healthy soil equals to healthy people. Healthy soil is public health. So therefore, everyone has right to have a healthy soil. Therefore, let us protect our soils. That leads to One Health concept. Thank you so much. Feed the soil, feed the future.
So I'd like to take this moment to give thanks to respected our for the lecturer who has given the top on the topic Microbial Technologies for Regenerative and Sustainable Agriculture. Thank you, KRK Reddy sir, for such an informative lecture. Our first year BSc Agriculture student has also joined this lecture, and I think this lecture is equally useful and very inclusive topic. Thank you once again, sir. I would like to I'm thankful to our Vice Chancellor, Professor K.R. Sambashivarao sir, for arranging such a wonderful webinar. Thank you, sir. So now the forum is open for any queries. If anyone has queries, they can raise your hands or they can unmute themselves and raise the questions.
If there are no questions, thank you so much.
Thank you, sir. Thank you once again.
Dr. K.R.S. Rao is here. So thank you, the Vice Chancellor. Thanks for giving me the opportunity to speak to the students. Thanks a lot. So all the best.
Thank you, sir.